Don't break it.
All right, so this is Dr. Robert Young, and we're just reviewing the procedure of how to take a drop of blood from the middle finger. Generally, that's where it's taken from. How to then take or press that blood onto a slide, and then allowing it to dry in order to then view it at some later time. So what I've included is some of these what are called Kim wipes. And the reason why I use this tissue is because it's a very special tissue, and you don't get fragments when you're cleaning the finger off, and you're just taking off any surface particulates from off the finger. You don't want to clean the finger with alcohol, because if you clean the finger with alcohol, that's going to affect the blood when it comes in contact with the alcohol, and it will actually crenate or damage the cell membrane. So I use these to clean the finger off. And what I do is I start like with a peace sign, and then I turn it this way. Yeah. And from this first knuckle, I kind of hold it.
Can I do it while we're talking?
Well, yeah, you can practice. I don't want you to prick the finger yet, but yeah, you can just hold it. So once you wipe the finger off, then whoever is taking the blood where you're doing it, or is it Megan?
Yeah, I'll be assisting.
Yeah, I'll be assisting. You then take off this part. Now, this is the lancet. This is a one usage lancet, and you twist it until it pulls out, okay?
Okay. Now I have 4 of these.
The reason I gave 4 of you, because sometimes there's mistakes, and so you've got
no, but these ones, I have 4, right?
Yeah. So in this packet, this packet are slides and also cover slips. Cover slips is a very fine piece of glass, and you should try not to put your fingerprints on these, but try to handle them from the edges. Now, if it's not in the first packet, it'll be there, because I included several of these. There's 2 that includes the cover slips, and 2 that just have the slides.
That has cover slips, and these 2 are just slides.
So this is what we're looking for.
Why do we need cover slips?
Okay, so this is what the slide looks like, right?
Mm-hmm. Yeah.
These are sterilized slides, and whenever you're handling a slide, you want to handle it from the edges, or you can handle it from the frosted end. You see, there's a frosted end of the slide. You can handle it this way. Some people feel comfortable this way. I feel comfortable holding it from the side.
Should I clean it? Because I think we might have touched it with our hands.
If you touched it, you can then take your Kim wipe, that's what they're called, and you can dust it off a little bit just to take off any sort of particulates that may be there, micro or nano, or any fingerprint. Okay, because we want to get a true picture of your blood. So
got it.
So if we start, there's two actual tests that we're going to take blood for. One is for the live blood cell test, which is we want to be able to put blood onto the cover slip and then place that down on the slide. So you should take your slide and find a surface. I would put down one of these Kim wipes, just set it down on a surface, and then what you do once it's set down, you can then set your slides on the surface so that they're ready to be picked up when you're ready. So if I was going to do this, I'm just going to set them down here on the computer, and then I would set my cover slips nearby, you know, so that I can pick those up too. So you can set one or two of these Kim wipes.
How do we grab the cover? How do we grab the cover slips?
You grab the cover slip by the edges.
No, with the cover slip is the little one though, no? So
Yeah, the cover slip is the little one.
You don't put your full fingerprint on it. Okay. Okay, so this one. Okay.
Yeah, and these are very fragile. The cover slips are very fragile. So if you do clean those off, you just do it very lightly, you know, because if you bend them, they will break. Where the slides are more rigid. There's more glass there.
And you gave us 4 of these cardboard things, right?
Yeah, and the 4 cardboard cases contain 2 regular sterilized cells in each little pocket. So you have these little pockets here, and I've put 2 slides in each of those. So in each one of the pockets, you have a slide like this.
Mm-hmm.
And then on top of that, you have the cover slips on 2 of these. So you got 2 of them that have slides with cover slips, and 2 packets. If I open this one up, these just have the slides, if you can see that. Okay, so you can open those up if you want to look at them. And
yeah, we did.
Yeah, if you want, you can actually keep them in the packet, and then this little cutout here, you can actually pick them up. And I purposely put the frosted slide portion to the top of it, so you can actually put your fingers on that.
Great. We made a little bit of a mess.
Do you have any crazy? Do you have crazy glue?
I don't think so.
Okay.
Do I need that?
It's helpful, but if you don't have it, that's fine. Okay, so let me show you how you can do this, and I will be my own demonstration. Okay. So,
wait, I should do mine at the same time, right?
Yeah, we're gonna do 2 samples of live blood, and we're going to allow that blood to jump to the cover slip, and then we're gonna set the the slide cover slip down onto the slide. So in order to do this,
Anna, can you find me some crazy glue, please?
Some what?
Crazy glue.
Yeah, just any sort of.
I'm in Adina's room.
Yeah.
Thank you.
So
Bye.
here's the cover slip, and when I put my when my
which one's the cover slip? The really small, right?
Yeah,
okay.
When my blood is on the finger, I'm going to allow the blood to jump to the cover slip. I don't touch the blood with the cover slip. I'm going to allow it to jump to the cover slip. So
So if you put it close enough to the actual finger with the blood, it'll just kind of do it on its own.
Yeah, and the reason for that is because the surface of the slide, the surface here is positive and the blood is negative. So it attracts. So you don't have to touch the blood on this test. Okay. So then what you do is you take your lancet, your single lancet, and then you put this just not down here at the tip, not to the side, but just right here, not down by the knuckle, you put it right here, just on the finger pad. You see that?
Okay.
Yeah, so I'm not down here on the tip. I'm just on the flat surface where the knuckle is.
Yeah. So put it like right there. Good.
We have
Okay, yeah,
Gorilla
right.
Super
You're
Glue.
gonna put it right here on the flat surface of the pad.
We have Gorilla Super Glue.
Oh, good. So if you have the super glue, you need to have that ready to be able to, once you set the cover slip down on the slide, you're going to seal the edges around the cover slip so we don't allow any air to come underneath the cover slip and be exposed, the blood to be exposed to any oxygen. So we want to have a sealed, contained sample of the blood. So let me go ahead and show you how to take the blood now, if you're ready.
Yeah, should I do it at the same time?
No, don't do it at the same time. Just watch first, okay? Because I got to show you how to take the blood out. So I'm going to push down like this. I'm not just setting it there. I'm pushing down, and then I'm pushing this part of, I'm pushing this top part. That part will release a spring inside this lancet applicator, and I push it. Okay, if nothing happens, it means I need to that's not going to work. Okay, I got to take another one. That's why sometimes.
How many? Do I do this to only one finger, or am I doing this to like 4 fingers?
You're doing this just to one finger. Yeah.
But we're getting a lot of samples. Is that what we're doing?
Wow, this is not, for some reason I'm doing this wrong. Hold on.
That's blood behind him, by the way. That's a picture of blood. Didn't you say when you were talking to me that the blood cells of people that had COVID look different?
Yeah, it looks
Something
like
like
the
that.
blood behind me.
What is it, all splattered?
No, it's coming apart. The membranes are coming out. They're deteriorating, giving rise to appendages or crowns that come off of it. This is what normal blood should look like. Let me show you.
Spherical.
This is normal, healthy blood.
Honeycomb. My goodness.
This is.
Is there any way to fix it?
Not.
How do you
can't.
fix it?
Yeah, you fix it by changing the environment so that you have healthier blood. You're building healthier blood. Okay, so once you have that pin out, you press it down and you click it. Okay, once you've clicked it, you'll feel a pinch. That means I've penetrated the blood. So I'm going to just show you this. See, and I don't squeeze the blood out. I pull the opening open like this. And all I want is a tiny, tiny little BB of blood. Okay? And first off, once I have the blood there, I take the Kim wipe and I wipe it off. Then I pull apart again and just express the blood. As soon as I have the blood expressed.
Or why didn't you wipe it the first time?
Yeah, you wipe the first drop of blood off. Once I have the blood there, I then drop the cover slip. You can watch how this jumps. Can you see it? I can then.
Oh.
Well, I kind of went into it. And then I immediately set it down on the slide. So once the blood jumps.
When do you put the glue?
You put the glue on and you seal all these sides. You seal them after you put the blood onto the slide. So once the blood is in on the slide, you then seal the cover slip around that slide.
So to seal the cover slip, you just outline the border?
Just outline the outside of that.
Do you have to press it at all to make sure it kind of?
No, you don't have to press it at all. Don't press on the slide. Just outline around the edges of the slide to seal it
before you put it on.
Oxygen exposure.
To seal it.
Okay. I'll show you.
Does that make sense to you?
Yeah.
Okay, so let's look at this again. Once you have an opening, you can express the blood. Once the blood is expressed, like this, you
And
can
use a
use
little
a little bead of blood. You then take your cover slip and allow the blood to jump to the cover slip.
Okay. Then you'll put it on this and then seal it.
Okay, so once it's dropped on there, you can then drop the blood onto the slide. And this is how you do that. You take the cover slip and you set the first side down and then just let it drop down. Yeah. And if the blood is coming to the top, if it's coming to the top, you want to kind of let that blood kind of come down to the other side. Anyway, you want to get the blood inside the cover slip and spread out like this. So that's a nice spread on this is what it looks like. So I've got a drop of my blood. I don't know if you can see the blood there.
And so we're doing this twice.
Yeah, you're
Four
doing
times.
it twice. I want 2 samples, yeah.
But
Okay.
I have 4 books.
Well, I gave you the first 2 to practice, and if you do great, you don't need to use the other one. Okay?
But there's different names. Some say dried blood,
Right.
and the other one says live blood.
Okay, the live blood has the cover slips in them. That's for the live blood. I haven't got to the dry blood. So set the dry blood samples off to the side.
But so
And
the
let's
live
just-
blood you do need 2 samples of.
That's correct. That's a whole
Okay.
different procedure. I'll have to show you how to do that.
No, the live blood, that's the one you just told us?
Yes, we're just working with live blood, putting blood onto the cover slip, okay, and then onto
yeah.
the slide.
Okay.
But I don't need to
But 2
do.
different samples.
I don't need to do all 4.
No,
Well, we're gonna
he's
do
gonna
what
show
we're
you the
gonna
dried
do.
blood next.
If we do
No,
good,
this is
and
the
we
same.
get good samples, then we've got
It's-
I have a backup.
yeah.
I put in 2
So these
samples
2
as
go together,
backup.
and then this goes with the other one.
Or I should say 2
Okay.
packets.
So this one was in theory in case we totally screw it up.
Oh, okay. Because you screwed up.
Because you screwed it up. Because you're dehydrated today and you don't want to give us blood. Okay. All right. Good. Ready for the next one.
Okay,
Cool.
so you want to set these up now, or do you want to?
Yeah, I want to do it.
Sure.
Okay, so you can use the peace sign here and just let this fold your other 2 fingers back. You know, you can hold it with your thumb, and then when you're expressing the blood, you express it by pulling the skin apart. See, I've already clotted, so it's not working. But you pull apart rather than push. Do not push the blood to squeeze it out. You want to open the puncture wound so the blood freely comes out. And you don't want a big drop of blood. You want the blood to be the size of like a BB, just a small BB. So a small bead of blood.
And this is for only the live, not the dry blood,
That's
right?
correct. This is for the live. So in the live blood, we're going to look at these structures like behind me. See, we're going to look at the structures of the red blood cells and see what type of health those blood cells are in.
Is it mailed or the office is here?
We mail it back to you, right?
Yeah. Then what you do is you seal it, put it in so that it doesn't open. So these are for mailing. You can seal it, and then you can put it in a packet to send it back to me.
Well, how do you seal it?
there a
You
time
seal it
or
by
a
just
tray?
taking tape and taping it.
Oh, okay.
Yeah.
Is there a time limit as soon as we do it? Like how?
There isn't.
How?
Once you've sealed the live blood around the edges with the crazy glue, there's no time left. It's like time is stopped. It's like putting food in the refrigerator. It's basically not going to deteriorate. It's
Got it.
over time it will, but there's plenty of time here. With the dry blood, that's a whole different procedure, and I'll show you how to do that.
Okay. So let me do this first.
All right.
Let's do one step at a time. Okay. All right.
So
You ready,
open
Maggie?
off your finger.
I did my finger.
And then this guy.
So clean the
twists
finger off.
off.
The index finger drops down or back. So you either have to pull it back like this, or you can drop it down, whatever you feel comfortable with. Yeah, the other two fingers, yeah, that's fine. So right on the pad,
So
push down and click.
got
Okay.
it. Okay, so do
Now
not.
now
not
from
you
there,
wanna
what you do is you express
Wipe
the
one.
blood. You can
Mhm.
pull the wound
Well, he said
open.
wipe this one.
After. Okay.
Yeah.
So this is the size approximately that you would want.
Let
I
me
know we
see.
wiped this one.
I
We wiped
can't
this
see
one.
it.
Okay, hold on. Let me see.
That's too
Where's your
big.
finger? Now it's too big. Hold on, it's too big now.
So we
Okay,
wiped the first
so
one.
what you do is you wipe it off with the Kim wipe and you beat it up again. Okay?
Yeah.
So once you've beaten it up to a little tiny BB, go ahead and take your cover slip, bring it over the top of that blood and allow that blood to jump up to the slide.
I don't know if it's jumping.
Okay, well, go ahead and touch down lightly into the blood.
Oh, jump.
Okay.
Okay, now set that onto the
Is that
slide.
enough?
Is that enough? It looks really small.
Yeah, you need to set it down. That's fine. Yeah, put it down on the covers on the slide. Set it down on the slide,
Okay.
and the blood should then spread out once you set it down.
Got it.
Okay, let's see what you got.
She's a professional
It's
doctor.
Pretty big.
Okay, great. Okay, we're going to take a little less blood next time. Okay, that's just a little bit too much. It's okay. We can use that. Go ahead and seal it. Well, forget that. Let's take the second drop of blood. We can
Okay.
seal that. We have time here. Let's go ahead and wipe off the blood with the Kim wipe, and then beat it up again, and then take another drop of blood onto the cover slip.
That's good.
Okay, cover slip. Here we come. Okay, then drop that down onto the slide. Let's see what you've got now.
This one might be too small.
Okay, that one didn't spread out. So what you can do there is just tap to the center, just tap it a couple of times lightly and see if it spreads apart. Let's see now.
Let me see. There might be too much glare.
Not enough?
Move it, yeah. I think we need to take another sample. Let's try it again. So we've got that other packet. Let's try the first slide is okay. Let's see if we can try not to squeeze your finger or you'll damage the blood. So
Should we
if you've got
should
another
we
slide with a cover slip, let's see if
seal?
we can take one more.
It's here. It's here. It's here. It's here.
Okay. Do we need to re-prick the finger or?
Do I need a prick again?
If the blood's not coming out, see if you can open it up.
No, it is.
Okay. Just a tiny little BB. That's all you need. Okay. Once you've got the blood expressed, hang on, I'll be right back.
Okay. That's it. You did it, Maggie. That's a BB.
Is that BB?
I think so. I don't need more. You need more.
I think the last one was like that size. No, this isn't good for someone who doesn't like.
I don't think David's gonna do this so well.
Uh-uh.
Cry like
Oh.
a baby.
I just had a situation with a client here. Can I
That's good.
Should be it.
put you on hold here. I'm just going to end the recording. Let me just see what let me see how we're doing here with the next one.
Okay.
Look at it. Look.
It's
Okay, and then
tiny.
you just set that on top of the slide.
Oh,
And
good.
once it's sealed,
Good one. Best one ever.
yep, that's it. Okay,
Woohoo!
perfect. Okay.
And I'll seal them.
You can practice this. This is on the dry blood. And what you're doing there is you're picking up the blood by tapping. So on this test, what you do is once the bead is up, you let it sit for 30 seconds. After sitting for 30 seconds, you then take your slide. The slide should be leveled. And it's like landing that slide into the blood where you tap lightly on the finger. So it's down, up, down, up, down, up, over, down, up, over, down, up, over. So this is what it looks like.
Do I clean my finger?
You can, yeah, clean the finger. Clean the finger. I'm going to have to do one more tap here. So now I've got a drop of blood. Watch this very carefully. So with the drop of blood, I then bring the slide over here and I go down, up, 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 down. So you notice that the blood has about 7 to 8 drops. Okay, some are larger because that's the surface blood that you're taking. And you want to get at least 7 or 8 drops of blood on the slide, and you're going to pick up all the blood off the finger.
Now
So
the
go
blood that
ahead.
you're getting on the finger, is that from the same BB size, or you can
Yeah,
let it get bigger?
Yeah, just the same drop size of blood. Okay.
Can I do the other hand, or do I have to stick
You can
with
do
the
the
right
other
hand?
hand if you want, but you can use the same hand if you want. Or
No, I
you
don't want to. This one has a boo boo. You see?
Okay, so you can, yeah, so you can take it from the other finger. Okay. Yeah, see this one here, all you do once again is bead the blood up, just like that. See that nice little bead? You can see the size of it. Can you see the size?
Yeah.
And then when you're coming to pick it up, watch one more time. Watch how I pick it up, okay? Just like this. 1, 2, 3, 4, 5, 6, 7, 8. That's it.
Okay.
That's all you
And
do.
then set it aside to let it dry.
Then let it dry. Yeah, don't set anything on top of it. Allow it to dry.
Okay.
I'll be right back.
Okay.
You got this, Megan. Let's glue first. You want to glue?
Yeah. So that we don't forget. Crazy.
I'm just going to stop the recording, okay?
Okay.